Here we'll learn the components of a eukaryotic animal cell. To begin, draw a plasma membrane that has two layers. Use a zoomed-in window to show that each layer comprises a row of phospholipids. Thus, it's often called the phospholipid bilayer. As well, show that it comprises proteins, cholesterol, carbohydrates. They associate with the bilayer in a variety of ways. Write that the plasma membrane encloses the cell, providing a protective barrier from its external environment and regulates traffic of materials in and out of the cell. Label the external environment as the extracellular space and the internal environment as the intracellular space, otherwise known as the cytosol. The cytosol is the fluid portion of the cytoplasm that does not include organelles. Write that the cytosol is an aqueous or water-based solution that bathes the organelles and contains a variety of molecules such as ions, metabolites like fatty acids, carbohydrates, and amino acids, as well as proteins such as enzymes. These molecules make it more gel-like than liquid. Before we add organelles, let's learn about an important network within the cell. Write that the eukaryotic cell contains an endomembrane system, which is a select group of membranous organelles that regulate protein trafficking and metabolism. We'll learn its components now and then list them for our review. First, draw the nuclear envelope as a discontinuous double membrane. Label the gaps in the envelope as nuclear pores. Indicate that the nucleus lies within the nuclear envelope. The nucleus is the largest organelle in eukaryotic cells, approximately 5 micrometers in diameter, and visible under a light microscope. Write that the nucleus stores the cell's genetic material, DNA. It also is the site of DNA replication and RNA synthesis, transcription. We'll see that protein synthesis, translation, occurs in the cytosol. Within the nucleus, draw a representative thin strand of chromatin, the uncondensed form of DNA. A nucleolus. Nuclei contain one or more of these non-membranous organelles. Cells that produce a large amount of proteins have a proportionally large nucleolus and a large number of ribosomes. Draw a strand of rRNA within the nucleolus where it's synthesized. Ribosomal proteins assemble in the nucleolus before maturing in the cytoplasm. Next, draw several free ribosomes suspended in the cytosol. Using a zoomed-in window, show that each ribosome comprises two subunits, one large and one small. Write that a ribosome binds to mRNA to synthesize proteins via translation. The ribosome can be suspended in the cytosol or bound to the endoplasmic reticulum. Now draw an endoplasmic reticulum that's continuous with the nuclear envelope. It comprises membranous sacs and tubules called cisternae. Show that the cisternae enclose a space called the ER lumen. The ER membrane separates these luminal spaces from the cytosol. It's continuous with the nuclear envelope. Next, show a representative portion of the ER as covered in ribosomes. Label it rough ER. Bound ribosomes synthesize and secrete proteins either into the lumen where they fold into their native conformation or into the ER membrane to produce transmembrane proteins. Label smooth ER as the portions that lack ribosomes. Now write that the rough ER is a site of protein synthesis, processing, and secretion. Next, write that the smooth ER maintains the following functions: lipid synthesis, production and secretion of steroid hormones, carbohydrate metabolism, detoxification of substances. As a clinical correlation, drugs and/or alcohol can induce the proliferation of smooth ER in liver cells, which accelerates detoxification. Next, let's illustrate how the ER secretes proteins. Draw a transport vesicle in the cytosol. Show that transport vesicles bud from the ER. They keep secretory proteins separate from proteins synthesized in the cytosol. Next, draw a Golgi apparatus as a series of stacked cisternae. Label the side of the Golgi that faces the nucleus as cis. It receives cargo from the ER. Label the opposite side of the Golgi that faces the plasma membrane as trans. Proteins exit the Golgi from the trans face. So show that the transport vesicle fuses with the Golgi apparatus on the cis side. Write that the Golgi modifies, stores, and secretes proteins and lipids that it receives from the ER, and that it synthesizes its own lipids. Cells active in secretion have an extensive Golgi. Next, draw a lysosome, then an endosome. Write that a lysosome is a vesicle that contains hydrolytic enzymes synthesized in the rough ER and processed in the Golgi. Write that an endosome forms when the cell engulfs nutrients or other particles via endocytosis. Use an arrow to show that endosomes mature into lysosomes and digest their cargo. As a clinical correlation, Tay-Sachs disease is a lysosomal storage disease that presents when lysosomes are missing a lipid digesting enzyme. Lipids accumulate in cells because lysosomes cannot digest them. As a result, they impair brain function. Now, this concludes the endomembrane system. To review, write that the endomembrane system includes the nucleus, endoplasmic reticulum, Golgi apparatus, endosomes, and lysosomes. Now in the cytosol, draw the outer membrane of a rod-shaped mitochondrion. This organelle is considered the cell's powerhouse because it generates all of the energy, ATP, necessary for cell function. Using a zoomed-in window, let's draw a mitochondrion. First its outer membrane, and then show that the mitochondrion's inner membrane folds to form cristae. It has a double membrane, just like the nuclear envelope. Label the space within the cristae as matrix. Label the space between the outer and inner membranes as inner membrane space. Write that mitochondria synthesize ATP, the cell's energy currency, that they do so via the citric acid cycle and oxidative phosphorylation, which is the coupling of two reactions, the oxidation of nutrients and the phosphorylation of ADP. Mitochondria have electron transport chains and enzymes embedded in their inner membrane, which facilitates these reactions. Finally, show that free ribosomes lie within the mitochondrial matrix. For review, write that two organelles within the cell have double membranes, the nucleus and mitochondria. Now let's draw the remaining cytosolic features of the cell. Draw a peroxisome as a small membranous organelle. Write that they produce hydrogen peroxide from the detoxification or breakdown of various substances such as alcohol. Any leftover hydrogen peroxide is then broken down into water and oxygen by an enzyme called catalase. Peroxisomes do not bud from the endomembrane system like lysosomes do. Now let's draw the cytoskeleton, the structural support for the cell. Draw microfilaments, then intermediate filaments, and then microtubules. Write that the cytoskeleton anchors organelles and provides the structural framework for the cell. Show that the microtubules nucleate at a region called the centrosome. Indicate that it contains two small structures called centrioles, and that the whole structure functions in cell division. This concludes our diagram.